about to meet a genius. This is a man who knows an awful lot, not only about malnutrition in general, but about muscle strength in people with liver disease. And I'm very glad to introduce Dr. Dasarathy, who will talk to us about nutrition in PSC.
Thanks, Dr. Carey, and thanks to the organizers for giving me this opportunity to share with you some of the work that we've done on understanding nutritional consequences in liver disease and in patients with cholestatic diseases. So I want to start by saying that malnutrition has been identified to be the most common and frequent clinical problems in patients with liver disease, much more so in all patients with cholestatic diseases. It affects up to nearly 90% of patients, depending on the stage and severity of the disease. For the same severity of liver disease, patients who have cholestasis, which includes PSC, have the most severe form of malnutrition. There are two major components that we have identified, which include skeletal muscle loss or sarcopenia and a disorder in energy metabolism, both of which are common in patients who have cholestatic diseases and in patients with cirrhosis. So how do we measure muscle mass, and how do we define sarcopenia? I'm trying to find if there's a pointer in this. Anyway, you can see that there is a large number of methods that have been used, and we have evaluated a large number of them. And these include simple measurements, which are how much is the size of the arm, how much is the size of the thigh. There are also small instruments, there are also larger instruments. You heard about the DEXA for bone density. We can also use the DEXA to measure the bone mass. And increasingly, we are using CT scans as a method to measure skeletal muscle mass in the body. Now, the standard thinking in patients with liver disease is that they have malnutrition, which is primarily loss of skeletal muscle mass and muscle contractile function. Muscle is, to a large extent, protein. So if you want to replace muscle, you should feed them. And you should feed them using different strategies. Make them eat more. Put tubes into their nose. Put tubes into their mouth. Every study till date that has shown that in the long term, nutritional supplementations do not help patients with liver disease. Feeding them more, asking them to eat more, really does not work. So why do we study muscle loss in liver disease, especially in the background that we know that these nutritional supplementations are really not very effective? If you see the top left bar graph, on the y-axis are the percentage of patients, on the x-axis are the various complications of liver disease. And you can see that loss of muscle mass is the most frequent complication. Unfortunately, despite nearly universal recognition by patients and by their treating clinicians, very little has been done to try to understand how to reverse this process. Today, the belief is that liver transplantation is curative for liver disease. Unfortunately, if you look at the bottom two line graphs that we looked at a few years ago, if you look in the x-axis is the years over which the numbers were evaluated. On the x-axis are the number of patients in thousands or hundreds of thousands. The red line is the total number of transplants that are done in the United States. We have not been able to do more than about 62 or 6,300 transplants a year. The black line shows you the number of patients who develop cirrhosis. If you see, the number of cirrhotics keeps increasing, but the number of transplants had remained constant. So this results in what we call an inverse funnel effect, which is that over time, the number of non-transplanted patients is going to continue to increase. And the mainstay of their management is going to be to treat their complications. And I showed you on the top right that the major complication is loss of muscle mass. Even patients who get on the transplant list, if you see the line graph on the bottom right, the black line is the number of patients who are on the transplant list or waiting for 6 months or longer. And while they are waiting, they continue to lose muscle mass and they continue to have the consequences of muscle loss. So what are these consequences? Why do we spend so much time trying to figure out what happens with muscle loss? So I have taken 4 representative curves. Those of you who have a background in mathematics or statistics can understand that these are called Kaplan-Meier or survival curves. If you see the x-axis or the horizontal axis is the time over which the patients are followed up in months. On the y-axis is the probability of survival. What's the chance of a patient surviving? As the graph keeps dropping or the line keeps dropping, the probability of survival keeps decreasing. If you see on the top 2 line graphs, patients who have loss of muscle mass die more than patients who have relatively preserved muscle mass. I talk about relatively preserved because all patients with liver disease, especially those who have cholestatic diseases, have severe muscle loss. But if there is even some preservation of this muscle, their survival is better than those who have the most severe muscle loss, pretty much similar to what you heard for bone loss. But what is interesting is, unlike the other complications of cirrhosis, which reverse after liver transplantation, if you look at the bottom left, you see that patients continue to lose muscle mass after transplantation, and those who lose muscle mass die more. So patients start with a low muscle mass, they don't recover their muscle mass, and they die more as their muscle mass progresses. So how can we, so I have shown you evidence that muscle loss is important and that muscle loss is progressive and it affects survival. Now, it has no meaning unless we can show that improving muscle mass can actually improve survival. So in these studies, we showed that a procedure that was not done for muscle loss, it was done for something else. Another complication of cirrhosis, which is the development of ascites or fluid in the stomach and varices or blood vessels that can bleed. When we do a transjugular intrahepatic portosystemic stent, which is what the expansion of TEPS is, we find that a significant proportion of these patients actually regain their muscle mass. This is in fact the only intervention that I'm aware of that can actually reverse muscle mass in patients with cirrhosis. And when you reverse their muscle mass, their survival improves. So if you lose muscle mass, patients will die. If you reverse their muscle mass, they survive. Therefore, targeting loss of muscle mass should be a major clinical goal for all of us who take care of patients with liver disease. I hope that by now I have convinced you that muscle loss is critical in patients with liver disease. In the year 2006, there was an estimate made by the CDC that there are about 2.5 million cirrhotics in the United States. I cannot tell you what proportion is due to cholestatic diseases. I can only tell you these are the total numbers. There is an estimated 40,000 patients with cirrhosis who are added on every year. There are about 27,000 deaths directly related to cirrhosis. Muscle loss is a major complication, and it contributes to death, complications of cirrhosis, poor quality of life, and adverse post-liver transplant outcomes. Therefore, muscle loss should be a major therapeutic target in the management of patients with liver disease. I'm going to very briefly talk about what we have done over the last 22 years in trying to address this question. So to understand muscle loss, it is not in patients with liver disease. It is not enough if we understand liver disease. I'm a transplant hepatologist. What we need to know is how does the muscle mass is maintained by a balance between protein synthesis in the muscle, protein breakdown in the muscle, and a type of cell in the muscle that is able to help with the recovery process. There are a number of genes that regulate this, a number of pathways that regulate this. My lab has been very interested in this gene called myostatin. I'll talk to you why we are so interested in this. So the approach that we have taken is a comprehensive approach. So we take skeletal muscle from patients with cirrhosis. We have an animal model where we create a shunt in the portal vein. There are also others who are trying to use the bile duct ligated model, which is similar to what happens in cholestatic diseases. We also have generated a number of mouse models and cell cultures that help us understand the molecular mechanisms, why there is muscle loss in liver disease, and what treatments can potentially be helpful. So in sclerosing cholangitis, the circulating bile salts are increased. There is also development of the accompaniments of the liver cell dysfunction, which is that the body's disposal of ammonia is impaired, and there is a reduction in testosterone in men. Recurrent episodes of cholangitis results in sepsis and endotoxemia. There is fat malabsorption, which, if you recollect, is another component of malnutrition, which is a disordered energy metabolism. There is also vitamin D deficiency that you heard about, and these patients can get cancers, and there is a component of cancer cachexia that is added on to these patients. So it is not surprising, therefore, that muscle loss is a very major consequence in patients with sclerosing cholangitis. So one of the mediators of how the liver communicates with the skeletal muscle. You have liver disease. Why does the muscle respond to this? So for the last more than 2.5 decades, we have been investigating how ammonia, which is normally removed by the hepatocytes, is a possible regulator of muscle mass. So if you see the bottom graphs, you can see that the concentrations of ammonia in the blood and in the muscle in patients with cirrhosis is significantly higher. When there is liver disease, the muscle seems to try to compensate and does some of the functions of the liver. So what does this result in? It increases the expression of this gene called myostatin. You don't need to know anything. If you look at these, we don't need instruments, we don't need calculations. Healthy controls have a very low expression or level of this gene called myostatin, which decreases muscle mass. That's why it's called myostatin. Muscle statin blocks the muscle. And patients with cirrhosis have very high expression in the muscle. Subsequent to our reports, many other groups have now shown that in the blood it is increased, in other tissues it is increased. So what we did was we made a compound that can block myostatin, and we took our animal model in which we have an expression, increased expression of myostatin. If you see, the red line is the disease animal, which is like a cirrhotic animal, and the yellow bar is an animal that has undergone surgery so that it mimics what happens in the disease animal. And when we give them this compound that we made called follistatin, the rate at which the muscle makes protein becomes similar to what you see in the healthy animal. Correspondingly, the muscle size increases, muscle strength increases. So this is the first evidence that there is a potential target-based treatment, not replacing something that we think is deficient. Here we have a very specific abnormality. If we identify that abnormality at the cellular level, generate an intervention that can block that abnormality, we can actually see an improvement. Now, I've talked to you about the rate at which the muscle makes protein, but I also said that it's a balance between protein making and breaking down. So one of the pathways or one of the mechanisms by which the muscle breaks down protein that has been most extensively studied is called the proteasome system. If you see, there is really no difference in the proteasome system in the human cirrhotics versus the healthy control. So it doesn't look like this pathway is important. So what I've shown you in this cartoon is that myostatin is an important mediator of the loss of muscle mass. and the proteasome-mediated degradation doesn't seem to be a major player. So we then looked at another mechanism by which the muscle breaks down protein. This is called autophagy. And as the name suggests, the muscle tries to eat itself. This is because patients with liver disease are in a state of starvation. When I say starvation, I'm not talking about feeling hungry, saying the cells do not have enough energy, or they're not able to utilize many of the nutrients that are given to them. And this results in the muscle trying to eat itself. So in summary, what I've told you is that patients with liver disease have a reduction in protein synthesis and an increase in autophagy or self-eating. So the strategies that we have tried are to stimulate the synthesis of protein and decrease the cells trying to break themselves down. Once again, my concern is this comes back to the same strategy which has never worked. You observe an abnormality, you try to reverse it, you don't really go into details of why did it develop and how can I reverse it. So this is a very complex cartoon, but this is 25 years of my life. So what we did was we showed the mechanisms by which the skeletal muscle is lost in patients with liver disease. So you can see that there are a number of investigators from my lab who have helped me develop these. So we found that leucine, an amino acid, which is part of a protein, can help in reversing these abnormalities. So we did a number of cellular studies and animal studies, and we showed that if you give a large amount of leucine, it is possible to reverse muscle loss. So we did the first human study that had ever been published in human cirrhotics to show that if you give amino acids with a large quantity of leucine, it is possible to reverse some of the molecular and the protein synthesis abnormalities in the skeletal muscle in cirrhosis. I've talked to you about muscle size. Now I'm going to talk to you very briefly about muscle strength. Patients with liver disease have a reduction in muscle strength. They have decrease in contractile function. They complain of fatigue. All patients with PSC and PBC, one of their major symptoms is fatigue. I was very happy to see in that brochure that fatigue was placed number one amongst the symptoms. What causes this? It is a reduction in muscle size and a reduction in muscle contractile capacity. So what could be the reason? So we looked at the mitochondria which make the energy for the muscle. And I'm not going to go into the details of this, but all I'm going to tell you is that the muscle mitochondria do not function normally in a cirrhotic patient. So what treatment options do we have? How do we decide treatment? What options exist? Is reversal of sarcopenia possible, and does it affect outcome? What are the impact of the other medications that patients take? Again, I repeat, using a deficiency replacement is really not a good strategy. A mechanistic approach to identify the target and try to reverse the cause would be the most optimum approach. So there are a number of treatment options of which giving patients a late evening snack is the most effective and efficient way to prevent progression of muscle loss. All the other interventions that I talk about are still, unfortunately, experimental, and people in my lab have been working pretty hard trying to figure out and dissect these pathways. So we showed that cirrhotic patients are starving. So if you look at this bar, there's something called a respiratory quotient. The lower it goes, the more the body thinks that it is starved. Cirrhotics are in a state of starvation even after eating their food, which means they're not able to utilize their dietary intake. We also showed that as the respiratory quotient keeps dropping, and if you see the bottom left corner, which is where all the cirrhotics are located, you see that their muscle mass is less. So we can try to increase the respiratory quotient, which can then potentially move the muscle mass towards where we want it to be. So how does a late evening snack work? It shortens the period of fasting, and I encourage all patients to take a snack before going to bed, primarily a protein snack. I have no recommendations on which protein it is. It does not matter. Any protein taken around 8:30 or 9 at night before going to bed is the single strategy which mechanistically can stop progression of muscle loss. So this shows you how a late evening snack can change the way the body's biology works. And this study from a friend of mine in Australia showed that if you give late evening snacks, you can see the black bars, you keep seeing an increase in their muscle mass. What are we doing now for patients? There are actually no standardized protocols. We quantify their muscle mass, we sometimes quantify their muscle function, make all kinds of indices, all kinds of numbers, but without a treatment, without something that is effective, these will continue to remain predictive, somewhat like saying that your numbers are bad, so you're not going to do well. What should I do? Really nothing, because all we are spending our time is in figuring out what will be the outcome. I want to know what can I do. So what interventions exist? Nutrition and dietary supplementations, I just told you, are not particularly effective. If you want to do exercise, what exercise? Endurance exercise, which is running on a treadmill or an elliptical. Resistance exercise, weights, what should be done? Are they effective? We have spent a fair amount of time trying to understand the differential response of cirrhotics to exercise. And you must also understand that exercise increases the ammonia concentrations in the muscle. The muscle itself makes ammonia, and I just told you that ammonia is one of the mediators of muscle loss and decreased muscle strength. So is exercise really effective? So one of the most frequent complaints of both doctors and patients is exercise is difficult. There are no standardized recommendations. Why is it difficult? What is fatigue? What is an impairment in the inability to maintain their strength? How do we overcome fatigue? Unfortunately, I have only questions. I don't have answers. We do have models that we are working on. We hope in the next decade or so, it's not soon enough for all of you, but in the next decade or so, we hope that we will have answers that will help us reverse this process. There are also questions about what protein, plant protein, animal protein, dairy protein, how much, at what time, what frequency, how many calories, how do we overcome the unwillingness to eat, the lack of taste, and how do we measure a response to these interventions? So I want to summarize by saying that muscle loss has a high prevalence, it adversely affects clinical outcomes, it is not reversed by liver transplantation. The cause of the liver disease, stage of the liver disease, affects the severity and may affect the response to our intervention. We unfortunately have no treatments to reverse muscle loss at this stage. Deficiency replacement has been the approach, but it has clearly not been effective. I want to thank all these investigators. You can see, clearly I don't do much work. It's all these people who are around me who do all the work. I kind of just hang around and give some talks. So I'm going to leave you with this diagram that these are some of these abnormalities that we have been working on, and we hope that in the next few years we should have answers which will help patients to cause an improvement in their muscle mass and function. Thank you so much.